Good afternoon, everybody. Today I'm going to talk about the retrosynthesis of oxybuprocaine and ciprofloxacin. I'm going to start with the oxybuprocaine. First of all, I have to say some characteristics of oxybuprocaine. It has other names, benosignat or novesin, but its UPAC name is 2-diethylamino ethyl 4-amino 3-butoxybenzoate. It is used in ophthalmology and otorhinolaryngology as a local analgesic. Now I'm going to talk about the retrosynthesis. Our target molecule is the oxybuprocaine. We can identify two symptoms. After doing the carbon-oxygen disconnection, we have to find two possible synthetic equivalents. They are the 4-amino-3-butoxybenzoic acid and the DEAE. The last one can be separated into symptoms after a carbon-nitrogen disconnection. The synthetic equivalents are going to be oxyran and diethylamine. We are going back to the butoxybenzoic acid derivative. A functional group interconversion is going to happen. The amino group will interconvert to a nitro group. Then another disconnection happens, and two possible synthetic equivalents are the 3-hydroxy-4-nitrobenzoic acid and butan-1-ol. The first one is going to lose the nitro group, and then one of the hydroxy groups interconvert to a diazo group. This diazo group will interconvert to a nitro group, and then it is going to be removed. A benzoic acid is obtained. It will interconvert to a toluene. Finally, the methyl group is going to be removed to get a benzene. After doing this analysis, we can identify the possible starting reagents: benzene, butan-1-ol, oxyran, and diethylamine. Now we can propose a synthesis. First, we have to do a chlorination and an oxidation of benzene. The product will be benzoic acid. Then, a nitration will happen. The carboxylic group is a meta-directing group, so the nitro group will be there. This group will be reduced to amino group by zinc and water. Then this molecule is going to become a 3-hydroxybenzoic acid by deazoxidization and oxidation. Now a nitration happens, and the group nitro will be in para position because steric hindrance. Finally, the molecule is going to react with butan-1-ol with heat to 4-amino nitrobenzoic acid. The second part of the synthesis is the reaction between diethylamine and oxyran to get DEAE. Finally, the product of the first part will react with the one of the second part with heat, and we are going to get the oxybuprocaine. The ciprofloxacin has other names: siloxane, cipro, nofloxacin, but its UPAC name is 1-cyclopropyl 6-fluoro-4-oxo-7-piperazine 1-il quinoline 3-carboxylic acid. It is used to treat or prevent certain infections caused by bacteria like pneumonia, gonorrhea, typhoid fever, and infectious diarrhea. Now I'm going to propose a retrosynthesis of ciprofloxacin. First, a carbon-nitrogen disconnection is going to happen. A piperazine and fluoroquinoline derivative are proposed as synthetic equivalents. The piperazine is going to be a starting reagent. Then another disconnection happens. That breaks one cycle. Now a functional group interconversion happens. Another carbon-nitrogen disconnection. After some steps, dietoxymetoxietan is proposed as another starting reagent. The retrosynthesis continues until we get the last starting reagents, that are the ethyl propanoidioate and benzene. Now I can explain a possible synthesis pathway. The first step is going to be the chlorination of benzene to get chlorobenzene. Then nitration. All the halogens are para-directing groups, so the para-chloronitrobenzene is obtained. Then another chlorination. The new chlorine will be in ortho position because steric hindrance. Then unfluorination. Another reaction will happen to get this molecule. Finally, thionyl-chloride will be used to transform the hydroxy group to chlorine. Then the product of the first step is going to react with the ethyl propanoidioate, and eventually this molecule will be obtained. This one is going to react with the etoxymetoxietan to get this molecule that reacts with cyclopropanoamine. Finally, this molecule will be reduced with sodium hydride. This molecule will react with piperazine to get the target molecule, the ciprofloxacin. Now I will propose another retrosynthesis. It has some similarities with the other one, but there are big differences. For example, with this carbon-nitrogen disconnection, another starting ingredient is gotten. The bromocyclopropane. Also, the cycle is broken in another position. We can identify three more starting ingredients: the dimethyl propandioate, the methyl format, and the benzene. Now we can propose the synthesis. First, the nitration of benzene, and then a reduction to get aniline. The aniline will be transformed in fluorobenzene by a Balt-Schimann reaction. The fluorine is a ortho-para directing group, so the para-fluoronitrobenzene and the ortho-fluoronitrobenzene are obtained. We can separate them with some techniques, like crystallization or distillation. Then a polymation happens, and then a reduction of the nitro group in amino group. Now the dimethylpropandiate and the methyl format will react to get this molecule. The products of the previous steps will react with heat and this molecule is obtained. Then it will react with diphenyl ether, and with these conditions the cyclopropane is going to be added to the molecule. Finally, this molecule will react with piperazine and reduce with sodium hydroxide to get the target molecule. Thanks for your attention. I hope you enjoyed this video. Be sure to smash that like button and subscribe.